Yeah, how are you doing? So we'll be having another class today. So basically we'll be going into cell cycle and cell division, where we'll be discussing more about mitosis and meiosis based on our syllabus. But before we continue, I want us to look at this diagram. Now, you see G0, that's GO, G1, S, and G2. And after G2, we have the prophase, we have the metaphase, we have the anaphase, the telophase, and the cytokinesis. Now, from prophase to the cytokinesis stage, or to the telophase, basically, it is named the hem phase. But today, on this very for this very class, I'll be taking only the mitosis. So the next class, I'll be taking the meiosis. I know you actually have a lot of us have understanding about it, so we know what it's all about. But also, I'll just be, it'll be more of point, and for us to have better understanding based on the way the syllabus wants you to attend to it. Now, the G0, the G0, the G1, the S phase, and the G2 phase, they are all the interface. Because from the explanation of interface is assumed to be the resting phase. So we say it's a resting phase, but it's not a resting phase because a lot of things go on during interface, which we don't know, and it involves a lot of sentences. But before it synthesizes the materials that will be used for prophase, yeah, before it synthesizes it, there are actually some things that need to be in place. So you begin to see those ones in G0, the G1 phase. After we have the S phase, which is the synthesis phase, then we have the G2 phase. Now, the topics we'll be looking at, or what we'll be examining here, is we'll look at the phases of cell cycle, which are the interphase and the M phase. So the M phase is the main mitosis or mitosis phase, or we say mitotic phase. So the interphase, which is the G1, the S phase, and the G2 phase. So the G1 is the gap 1, the S phase is the synthesis, and the G2 is the gap 2. So the M phase, which we have the prophase, the metaphase, the anaphase, and the telophase. And the other side is meiosis, meiosis 1, meiosis 2, and significance of mitosis and meiosis, differences between mitosis and meiosis. So on this very topic, I would not be adding the questions, but under the meiosis, I would now have all questions being asked on mitosis and meiosis. So I will now ask it under the second lecture, which to be a form of assignment for every one of us to attend to. And I think I will be checking it, so I'll be able to be sure that you guys really get all I've explained here. So let's go straight to the main work for the day. Now, mitosis. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. Now, in mitosis, it means just one cell, a particular cell, a mature cell, divide into two. Basically, the mature cell, most cases, they are parental cells. And that's why I said they would have been fully grown. So they divide into two, and once it divides into two, they form two daughter cells every time they divide. Now, a very good example of mitosis is the binary fission. binary fission that occurs in amoeba. When amoeba grow in its environment, it grows to a particular stage, that's the parental stage, and it begins to undergo cell division. So that's an example of what mitosis. So the cell cycle, let's see how we go about it. I don't want us to waste much time on it, but just get the understanding how mitosis occur. Are you getting me? Parental cell divide into two to form two daughter cells. And the funny part is that the daughter cell will have the same number of chromosomes as the parent, because the parent cell having the somatic cell divide into two, and the daughter still have a complete somatic cell. So a very good example of mitosis, when you talk about mitosis, is when we relate it to asexual reproduction or asexual reproduction. In asexual reproduction, the the parents will give birth to their to new offspring that will have the same number of chromosomes, so which means it doesn't really involve sexual reproduction. So it's going to be asexual reproduction. So we'll still talk more on that, just a little digression. So cell cycle is a sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells. Now, we talk about um, sequence of events. Now, if you look at the diagram beside it, those, that's an example of sequence of events that occur before there will be synthesis of more constituents of the cells. Now, when we get to cell, what's it called? To cell itself, the full topic on cell itself. So you will know what the cell is made up of. Now, inside the cell, there are a lot of what's it called? Organelles. Yeah, there are a lot of organelles that make up the cell. So, and every of these organelles have their own function. During reproduction, there are some organelles that must be very, very active and that must be readily available before cell occur. So that's why we say events by which a cell duplicates its genome, all the constituents of the cell. It synthesizes the other constituent of the cell, all the constituent of the cell, what the cell needs to undergo reproduction. And eventually, it will divide into two daughter cells. So I hope you understand that. So if you don't understand that, just you can just comment here and just tell me to um shed more light on that. I'll be able to do that. But basically, it's still what I just explained earlier. So physics of cell cycle. The cell cycle is divided into two basic phases, which is the interphase and the M phase, which is also called the mitosis phase. I've explained that earlier. So this is a cell cycle now, indicating mitosis. Okay. Then M phase, talked about that earlier, so I might just keep some things, especially when I've talked about that. So the M phase actually represents mitosis, which is the cell division, the process, and the interphase represents the phase between two successive M phases. Now, two successive M phases, that's mitosis phase, is the interphase. So we call it the resting phase. So interphase is called the resting phase. So we assume that when one division has occurred, the cell will rest, then another what's it called? Cell division will occur. So it's assumed to be the resting phase, but it is not actually the resting phase because there is production. Organelles are active, and organelles needed for a section of for mitosis. We continue go into synthesis. Now, karyokinesis. What is karyokinesis? Nuclear division corresponding to the separation of daughter chromosome. Basically, another way to say it is karyokinesis is the nucleus division. When the nucleus of the cell is undergoing division, so that is what we call what karyokinesis. So nuclear division corresponding to the separation of daughter chromosomes. Remember, chromosomes are found inside the nucleus of a cell. We have chromosomes inside the nucleus of a cell. Although we have three types of chromosomes. We are messenger RNA, what's it called? Messenger ribosomal and transfer RNA. Yeah, we have one which is the messenger, which is outside. So the other ones are inside. But basically, nuclear division is actually the process whereby, karyokinesis rather. Karyokinesis involves what? The division of the nucleus. Nucleus dividing, that is karyokinesis. Now, the cytokinesis, from the word cyto, cyto means what? Cytoplasm. Are you getting me? So the division of the cytoplasm is what we call cytokinesis. Karyokinesis, karyo is a word for nucleus, nucleus division. So that once you see that, you quickly know where you are going to in short answer questions when you come across it. Now, the M phase starts with the nuclear division. The mitosis phase starts with what? Nuclear division, corresponding to the separation of daughter chromosome, which is karyokinesis, and usually ends with division of cytoplasm, which is cytokinesis. Now, what are we trying to say? When cells are going through cell division, firstly, the nucleus starts dividing before the cytoplasm. I want you to take note of that. It's very, very important. So that when you want to explain binary fission in amoeba, you'll be able to say the nucleus starts dividing first before the cytoplasm. Now, let's go straight to the main point. Interphase. So the interphase is called the resting phase. So the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. Now, that's what I explained to you. We call it the resting phase, but it is not resting because work is being done during this process. So we say the cell is undergoing division and it is producing more DNA, replicating the DNA inside in an orderly manner with what? With cell growth. And that is why the cell needs to grow to a particular size before this can occur. Now, it is divided into three phases, which is what? The G1 phase, the G2 phase, and the S phase, which I've talked about earlier. Now, the G1 phase. What happened inside the G1 phase? Now, G1 phase is the interval between mitosis and initiation of DNA replication. The space, the interval between mitosis itself, that's when mitosis has occurred. The interval between mitosis itself and initiation of DNA replication. Now, like I said, we said interphase is a resting phase. Immediately after mitosis has finished occurring. Now, there would before we start the initiation of the DNA replication, the space between the mitosis and the initiation of the DNA to start replicating and growing and producing more of chromosomes and every other thing that is being needed for another cell division. This interval is called the G1 phase. Now, cell is metabolically active and continuously growing. There's continuous growth. It's not sleeping. It's not resting. Growth is occurring, but it seems as if nothing is happening in the cell. It does not replicate its DNA. It does not replicate its DNA. So there is initiation of DNA replication, but there is no replication. It does not replicate the DNA. Now, the S phase or the synthesis phase now, it marks the period during which DNA synthesis or replication takes place. Now, synthesis now, we now have replication. So it's in the synthesis phase that replication will take place. Okay. Thank you. And during this time, the amount of DNA per cell will double. The amount of DNA per cell will double automatically during this phase because we are talking about sentences like production, multiple production. So there will be what? Um, the DNA per cell will what? Double. Now, DNA replication begins in the nucleus. Centriole duplicate in the cytoplasm. The central in the cytoplasm, when we talk about what's it called? When we discuss about the cell as a whole, you get all these organelles, the central, the cytoplasm, the nucleus, the mitochondrion, and so on and so forth. We talk more about that. So, but all the same, that doesn't disturb you from listening to this before the cell. It doesn't still make any difference. Yeah, because we don't really have much terms to discuss about here. So we just basically talk about organelles that are responsible for reproduction. Those are what we'll be talking about here. So by the time you have your notes to cell, a full cell now, you'll be able to understand what each of them is used for. So the centriole duplicates in the cytoplasm. So the centriole is inside the cytoplasm, while the DNA is found in the nucleus. Now, the G2 phase, protein synthesis and cell growth continues. At G2 phase now, there is more protein synthesis and the cells still continue to grow. The cells that do not divide further exit G1 phase to enter an inactive stage called quiescent stage G0 of the cell cycle. Now, protein synthesis occur in G2, and at G2, the cells that do not divide, that do not undergo continuous division now, will now exit back to G1 phase. And from G1 phase to G0 phase, since those cells want to rest, they will give them time to go there and go and rest. Are you getting? Because the G0 phase is what's it called? Um I can say a full resting phase, like inactive, nothing is happening there. So cells in this stage remain metabolically active, but no longer proliferate unless called on to do so, depending on the requirement of the organism. So, like I said, it's more like the cells want to rest. So they will transfer them from G2 back to G1, back to G0 to go and become inactive there for a period until when the cell needs them. It's not like they are dead, just that they are what they are metabolically active, but they are in their what's it called? They are in their inactive stage. So once they are being needed by the cell, then they will be called upon. Then the whole thing will continue again. Aha. So in animals, mitotic cell division is only seen in the diploid somatic cells. Diploid somatic cell. What are somatic cells? Somatic cells are called the body cells. The body cells has 46 number of chromosomes. Now, the way we are now, the cells we have is called a somatic cell. Our body cell is called somatic cell. Our sex cell, our sex cell now is called sex cell. That you understand what I'm trying to say? That one is a sex cell. The body cell is called the somatic cell. So mitotic division basically occur in somatic cell, diploid somatic cell. Diploid somatic cell, meaning they have two N, 46 number of what chromosome in human being, for example. Now in plants, mitotic division occurs in both haploid and diploid cells. This can be one of the differences between animal cell and plant cell. So we should take note of that. Are you getting me? Aha. So mitosis occur only in diploid somatic cells. That is cells that have their full number of chromosomes. I don't want to digress because there are a lot of examples I can begin to cite. I don't want to go deep into those sides because they are also topic on their own. So when we get there also, we'll talk more about that. Now, in plants now, mitotic division occur in both haploid. Haploid means half number of diploid. Are you getting me? So it occurs in both haploid and diploid cells. Now, that's what we call alternation of generation in plants. If you have heard about alternation of generation in plants, so it means the haploid stage and the diploid stage both can undergo mitotic division. So I will still take you through plant too. That's botany, as we are doing in zoology. I want to quickly go through the whole zoology, so we have the whole information on zoology, and I'll now come to the part of botany also. So we'll be able to explain more on this mitosis under the plant. So in plants now, we will talk about it. It occurs in both haploid. Haploid is more like talking about the sex cell. So it occurs in both sex cell now and the somatic cell. That is what we are trying to explain here. Now, we go to the M phase. So the M phase involves a major reorganization of virtually all components of the cell. Now, at the interface, once there is production of this, synthesis of this, and everything is ready, now, the M phase now, which is the mitotic phase, we now begin to do a proper reorganization so that everything will align the way it wanted to align to form a new offspring or to form new offspring. Mm. So, like I said, number of chromosomes in the parent and the progeny cell is the same. I've explained that also. The number of chromosomes that the parent will have and the number of chromosomes that the offspring, which are called the progeny cells, they will have the same, it will be the same number of chromosomes. So if the mother is having 46 number of chromosomes, the child also will be having 46 number of what? Chromosomes. So it is also called an equational division. Equational division. It's also called that. We mentioned something like that under the topic we had before this. Can I just remember? Embryology. Under embryology, we talked about that equational division. So that's just that. But basically, what we are trying to say is the parent divide and form two new progeny cells, which we have the same number of chromosomes as the parent. That is mitosis for you. Now, mitosis is divided into 4 stages. We have the prophase, the metaphase, the anaphase, and the telophase. Now, the prophase now, the stages in prophase. Condensation of chromosomal materials. Remember, in interphase, chromosomal material, DNA, proteins, everything has been synthesized. So in prophase now, there is condensation of chromosomal material. Now, chromosomal materials become untangled during the process of chromatin condensation. Now, it's looked at it's tangled before, like you cannot see it clearly earlier. But now in prophase, you begin to see them clearly because chromatin material is being formed. Chromatin material are getting formed gradually. So you begin to see them gradually. It becomes untangled during the process of chromatin condensation. Now, the centriole, if you remember the centriole, we mentioned centriole there, that is found in the cytoplasm. So we say it's found in the cytoplasm. So now it's coming into being active. It's coming here to perform its function now, starting from prophase. Now, the centriole move toward opposite poles of the cells. So the centriole begins to move towards opposite poles of the cell. One at one side, the other one at the other side. Okay, now cells at the end of prophase. The cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, that's Golgi bodies, no endoplasmic reticulum, no nucleolus, and the nuclear envelope. So what are we trying to say? Remember, it's a whole cell on its own earlier, a whole cell on its own. But once prophase begins, and the late prophase, that's meaning towards the end of prophase, you will notice that what all the organelles, all these organelles that are supposed to be inside the cell, everything has what disintegrated, disappear. We don't have them again at the end of what the late prophase. Why? Because we don't really need them. Those ones we need basically are the ones that are responsible for what mitosis, cell division. Now we go to the next one, metaphase. Metaphase, disintegration of the nuclear envelope marks the beginning of the metaphase. Now we talk about nuclear envelope. Nuclear envelope is the one that surrounds the what's it called, the nucleus. Now, if you see, this is the nuclear envelope here. At late prophase, as you can see, it is what is disappearing. Now, at metaphase, now there is disintegration of the nuclear envelope. So it marks the beginning of what metaphase. So once you see that the nuclear envelopes has disintegrated, which means we have started what metaphase. Now, chromosomes are spread through the cytoplasm of the cell. Chromosomes are now spread through the cytoplasm everywhere, through the cytoplasm. Why? Because there is a job for them to do. They need to start coming together and, forming a full chromosome. I get me, that can be easily seen. Now, chromosome condensation is completed. At metaphase now, the chromosome condensation is now complete. So you can see that this one is chromosomes, not more chromatin materials. The chromatin material mean they are just strands everywhere. You don't really understand what the strands are. But once the chromatin material now join together at the centromere, then you now have what? Chromosomes. Well condensed, and you can see it clearly. Now, chromosome is made up of two sister chromatids. So when you talk about chromatids, you talk about chromatin materials, we are talking about the same thing. So chromosomes, now how is chromosome formed? Chromosome is now formed when two sister chromatids. Why do we say two sister? It means they have the same, they are similar in size and shape. So the two sister chromatids now come together and they will be attached at the center. They will attach themselves at the center here. That point where they are held together is now called the centromere. It's called the centromere. So if you look at this diagram now, you will see this is chromatid, another chromatid, and they come together and they join together at the centromere. Then we call them what? The chromosome. Okay, thank you. Now, kinetochore. Kinetochore, that's a small deep structure at the surface of the centromere. It serves as a site of attachment of spindle fibers. So remember, kinetochore is different from centromere. I've shown you what the centromere does. Now, for kinetochore now, they are small disc-shaped structure, They are found at the surface of what? That centromere that we talk about. The centromere that join two chromatids together to form chromosome. Now, you will now see the kinetochore on them, the book-like shape structure. What is its function? It serves as a site of attachment of the spindle fibers. Now, spindle fibers are also needed for migrations. So for the migration to be effective, there is a need for kinetochore on the chromosome, especially at that side where you have the centromere. So please, if they ask question about centromere and kinetochore, they are two different things. Centromere is the point where two chromatids meet and attach together. That's centromere. But for kinetochore, it is a disc-shaped structure that is found on the surface of the centromere. What is its function? It is a site of attachment of spindle fibers during cell division. Good. Now, kinetochoresis. This formed by the spindle fibers. It is formed by the spindle fibers. It allows chromosome to move into position at the center of the cell. Now, if you notice all this, these are the spindle fibers. So that is what we are talking about. The point where you have this is kinetochore. Kinetochore at the surface of chromosome, kinetochore at the surface of chromosome, kinetochore at the surface of centromere rather. You understand? So this formed by the spindle fiber, it allows this chromosome now to begin to move into position at the center of the cell. Now, this is what I'm trying to explain. These chromosomes that are scattered everywhere, the spindle fiber now with the aid of kinetochore on the centromere now. Are you getting me? It will now be as if it is drawing them to a particular position. Everyone that is scattered everywhere, it will now begin to what? To bring them to the center. Come around to the center. So it will bring everything, which are the chromosomes. As you can see, look at the chromosomes now. It brings them to the what? To the center of the what? Of the wall. Now, the key feature of metaphase are spindle fiber attaches to kinetochore of chromosome. You talk about spindle fiber anytime you want to talk about mitosis. Very important. Chromosomes are moved to spindle equator and get aligned along metaphase plates through spindle fiber to both poles. As you can see, there will be movement towards the spindle equator. Those are the features. Those are the important things that need to occur in metaphase. And at anaphase here now, remember, chromosomes are at the equator now. Then there is a need to now divide them. So at anaphase, each chromosome arranged at the metaphase. The chromosomes now that are at the metaphase are at the equator, at the center now. Split simultaneously. They will split simultaneously. Two daughter chromatids. Remember, two chromatids come together at the centromere to form the chromosome. Now, those chromatids will now what? Separate. The chromosome will what? Separate. Then you will have two daughter chromatids. It's not referred to as chromosome. We don't call it chromosome again. We call them chromatids. Remember, chromatids form chromosome when they come together. Now there is a division. Now referred to as chromosomes of the future daughter nuclei, we now begin to migrate towards opposite poles. It's very, very obvious. It's very, very obvious at this diagram here now. You will see how they now begin to migrate towards opposite pole. One move towards this pole, the other one move towards the other pole. Now, as each chromosome moves away from the equatorial plate, the centromere of each chromosome is moved towards the pole and ends at the leading edge with the arms of the chromosome trailing behind them. As you can see, so as it is moving towards the equatorial poles, these ones also are what? They are moving. As they move away, you notice what? The centromere is also, you see the centromere, which is at the edges, also move with them. centromere of each will move with them towards the edges where they are going to. Now, what are the main characteristics or events, key events that occur here? Now, the reason why I'm giving the key events is with the key events, you can summarize what is happening in each of the phase or each of the phases. Once you remember spindle fiber, once you remember moving towards the equator, you know what we are talking about. Now, once we talk about moving towards equatorial plates, you know what we are talking about. When we talk about moving towards the center, you know what we are talking about. So now, the key events here, the centromere split and chromatids separate, main events. And the other one, sorry for that. And for the other one, we have the chromosome. Okay. Now, for the other one, we have the chromatid move to opposite pole. The chromatids move to opposite pole. They move to the opposite pole, as you can see. The main key events that occur there. So it makes you remember anytime you want to talk about them, especially in examination. They ask you to explain mitosis. Write short note about mitosis, so you'll be able to explain all these things. Now, the next one is the telophase. What happens at the telophase? Chromosomes that have reached their respective pole condenses. Now, they've now gotten to their respective poles now. They now begin to condense and they lose their individuality. Remember, they look separate. They are separate earlier. But now at telophase, once they get to where they are going, they now condense. Everything now become one. Like a mixing together. It's now become what? One. So they will lose their originality. Now, the individual chromosome can no longer be seen, and chromatin material tends to collect in a mass in the two poles. Remember, I once mentioned this chromatin material. So they are the chromatin material that form the what's it called? The chromatids. Before the chromatids now come together to now form the chromosome when centromere join them together. Now, since they've gone into wherever they are, they will lose their originality and they now condense together and they become what? A chromatin material, which will be a form of mass liquid at each end of the pole. So this is what I'm talking about here and here now. Now, the stage shows the following events. What are the events that occur? Chromosomes cluster at opposite spindle 1 and 2, spindle pole, and their identity is lost as discrete elements. So they become chromatin material. They don't have, you cannot call them chromosomes again because they don't look like chromosomes, but you can call them what? Chromatin material. Now, the nuclear envelope assembles around the chromatin cluster. You see the what? You see the nuclear envelope. So nuclear envelope is just like, I think I use this explanation most times so that I know a lot of students really quickly understand it. It's just like when you use condom to hold palm cell, like you don't want it to peel off or to spill out. So you use it to hold it in place. That is just the same way you have this nuclear envelope. It wants all the nuclear granules. It wants everything to be in order, to be in one place and not scatter around. You understand? So there's a need for nuclear envelope to now begin to surround it. Now, at this part now, you will now notice that what the nucleolus, the Golgi complex, and the ER will be what? Reformed. It will now be reformed at this stage. Now, what are we trying to say now? All we are trying to say is the telophase is now the last phase. As you can see, the nucleus has divided already. That's why we always say karyokinesis occurs first. That is, the nucleus divides first before the cytoplasm. Now, this is the cytoplasm. This one has the nucleus has divided already. Then you have the cytoplasm now what? Dividing. Now, from telophase now back to the interphase, which we call the resting phase, which means we are now starting all over again. So the all over now means each of these will now begin to grow, 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 grow. When they now get to that stage again, they will now start all over to divide. And that is what mitosis is about. But we want to talk about some key things here. I want to talk about some key things so that we have more understanding on them. Now, cytokinesis, which I called cytoplasmic division. I've talked about that before. I think I've explained cytokinesis. So mitosis accomplish not only the segregation of duplicated chromosome, not only the chromosome now that is segregated now into daughter nuclei, which is karyokinesis. Not only karyokinesis that occur, but the cell itself is divided into two daughter cells by a separate process called cytokinesis. Now, if you just go through everything we have here, it's just explanation of what I started with, so you'll be able to have the idea. Now, at the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. Are you getting me? So everything that is needed in the cell will now be what? Will now be produced and it will be distributed. Now, in some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of phycitium. That's a very good example is endosperm in coconut. Now, all this is just what's it called? A little digression also. So we just want you to know that as we've earlier said that immediately after nucleus division, which is karyokinesis, cytokinesis occur immediately. But we are now trying to tell you some exemption. And a very good exemption is what the endosperm in coconut, the liquid endosperm in coconut. Are you getting me? Aha. So it does not occur immediately, which as a result of which multinucleate condition arises, leading to the formation of syncytium, which is what we call here. So that is just an exemption to what we discuss, so that you will have the idea that it's not in all cases, but basically in most organisms, both plant and animal, most cases you have cytokinesis and karyokinesis. And IGMB and JPEB also, they will also ask you what is the difference between cytokinesis and karyokinesis, differentiate between them. Anytime you see cyto is from the word cytoplasm, cytokinesis, division of the cytoplasm. Karyokinesis, division of nucleus. Now, what are the significance of mitosis? Mitosis, or the equational division, is usually restricted to the diploid cells only. We mentioned that earlier. But in plants, we have it in haploid and in what? In diploid. And also in some social insects, haploid cells also divide by what? Mitosis. In some social insects, I think you are aware of social insects, like termites, social insects. So in some of them, I'm not saying a particular one, in some of them, you might have haploid cells to undergoing mitosis, but basically it occurs most in diploid cells. Now, mitosis results in the production of diploid daughter cell. I've mentioned that too. And they will be identical in their genetic composition. I've mentioned that too. So it is going to produce a diploid daughter cell that will have the same number of chromosomes as the mother. Now, the growth of multicellular organism is due to what? Mitosis. Growth of multicellular organism, as I am now, I am an example of a multicellular organism. Our growth and our development, growing. This is not how I am 10 years ago. This is not how I am 20 years ago. So for my cells to multiply, produce new cells, for me to be tall, for me to be fat, and all those things, everything is by what? Mitosis. Because I have a somatic cell which produces another somatic cell, and that is how it continues. Now, cell growth results in distribution or distributing the ratio between the nucleus and the cytoplasm. Now, as I am now, a multicellular organism, for example, if growth is going to occur, it is this cell, this mitosis that we talk about, that will be responsible for equal amount of nucleus and equal amount of what? Cytoplasm. So it's not like that there will be more cytoplasm than more nucleus. No, it will give the exact ratio that is needed to form a new cell. Now, a very significant contribution of mitosis is cell repair. Yeah, that's also an example. When you have cut in your hand, and after a while you notice that what the whole place is covered up, and you are feeling like how you just say a repair happen, it's because eating protein. It's not just that is the function of mitosis. The cells around will begin to produce another new cell by mitosis, and they begin to what? To hold to each other and continue to form until they cover up the whole space. That is cell repair, by one of the significance of mitosis. And lastly, the cells of the upper layer of the epidermis cell, of the epidermis, cells of the lining of the gut and blood cells are being constantly replaced. Now, example of epidermal cell, epidermis, is the cell on our skin. Are you getting me? You will notice that sometimes, okay, under our foot also, for example, you notice that sometimes the upper part will peel off, another one comes up, and you understand what I'm trying to say. So peeling off and another one being produced, that is also the function of what? Mitosis. The lining of the gut, our gut also, lining of our gut. So the same thing, the blood cells too, because cells being constantly replaced, we see some of us that goes to donate blood. You donate blood, and within how many days your blood is back again. Like here now, I have a friend who I think almost on a monthly basis, he will always go and donate blood. Now I ask him like, why are you donating? He say, if he doesn't donate it, it's always affects him. So there are people like that. It will be as if blood is too much or something in their body. So when they donate it, new one is being formed by what? Mitosis. And the last point here, mitotic divisions in the meristematic tissue. The apical and the lateral cambium result in a continuous growth of plants throughout their life. So that is also an example. I don't really want to talk much about plants here because under plant also, that's under botany, we might still talk little about it, but all the same, it is still the same explanation that we have here. The same explanation we have here is what we also have here. So meristematic tissue are continuously growing tissues, and we can find them basically at the apical part and apical and the lateral part. Apical is the shoot side, the lateral towards the root side. So which means you see more roots coming out, penetrating, and for the apical, at every tip of every plant, you will see new shoots coming out, new branches, and so on and so forth. You now begin to like, how is it happening? That is mitosis, because new cells are being formed, and as new cells are being formed, the plants begin to what? Grow. So thank you very much. That is all on mitosis. Like I said, I'll be taking only mitosis now. Then I'll take meiosis also, because I want you to digest it properly and have a better understanding of of them. And after this class, just try as much as possible to summarize, like sit down and summarize it. Summarize it. Go to your past question, check questions there and summarize it. And if you don't have past question, like I told you, all the questions that have been asked in IGMB and some from JPEP too, or many from JPEP too, I would put it under meiosis. So you'll be able to see how they ask question on both mitosis and meiosis. Thank you very much, and have a blessed day, everyone. Thank you very, very much. God bless you.